Hi all. So yesterday, as you know, we were talking about ecosystem studies, and we want to continue with that today. We're probably going to finish up now today with the different sampling equipment that we use in ecology, and then the next class then we'll be looking at the ecosystem overall. So where we stopped off yesterday was we were looking at these qualitative and quantitative surveys that are done in ecology. Okay, so a qualitative survey is basically this where you want to see if something is present or absent. So let's say we're looking at rare species, okay? So for example, there is a very rare species of tree called the strawberry tree that is only found in County Kerry, okay? When I'm doing surveys in a particular woodland or grassland ecosystem, maybe down in that region of Ireland, I might want to use a quadrat to record how abundant that species is in that locality. So a qualitative survey simply will record if that strawberry tree is present or absent. And that is dependent on you doing a number of different surveys. So if I was doing a qualitative survey, it would work like this. If I wanted to get something called percentage frequency, what I would do is I would take out a pen, remember, I would throw it over my shoulder to make this random. Where the pen lands, I put the quadrat down, the center of the quadrat, and I will then note if the strawberry tree is present within my quadrat. Okay, now might be the strawberry tree. It could be something else as well. It could be, for example, I don't know, maybe nettles or something, or maybe some invasive species like a hogweed that is invading Ireland. You might want to record that the presence of that. So anyway, going back to what I was saying, you will repeat this process 10 times. In other words, so that you have recorded if the species was present in 10 quadrats. What you do then is you will have a table with ticks and X's, and you will tell from that basically how many of these species were present in each of the quadrats. Okay, so what we see here, for example, this is an example done out for moss. We can see here moss was present in the first quadrat, the second quad, or sorry, hold on, no. I'm hardly able to see there with the icons coming up. There we go, down the bottom. So we can see that the moss was absent rather in the first quadrat, absent in the second, third, fourth, fifth, but it was present in the last sixth, seventh, eighth, ninth, and 10th quadrat. So therefore, what we'd say, it was present for a total of 5 quadrats, 5 out of 10, multiplied by 100 over 1 will give you 50%. So that's a qualitative survey, guys. A quantitative survey is a little bit different. Quantitative, maybe we want to know a little bit more about the abundances of different organisms, okay? And in that circumstance, we'll do a quantitative survey because this actually records the numbers. So it's actually very simple, right? So again, we'll be probably doing this towards the end of the year, and when it's nice and sunny, hopefully, and not as miserable as it is outside today. What you will do, instead of taking maybe one of these quadrats here, you'll take out a quadrat that contains 100 squares, okay? 100 grids. And what you need to do is this. Repeat the steps to make it random. So you need to chuck a pen over your shoulder, and you need to put down the center square over the pen where it landed. Now, when you're measuring here, what you want to do here is follow the same procedure for each of these different grids. So this is the procedure. Say for argument's sake, I want to see the abundances of 5 different organisms. So let's say I want to look at grass, dandelions, daisies, buttercups, and moss. What I will do here is, if one of these species, we call it a hit. If it touches the top right hand corner of each of these grids, it will be recorded as 1. Okay, so say for example in this one here, grass. If I notice here that grass is touching the top right hand corner of each of these grids except for 5 of them, what I would say is that there is 95% grass coverage within the quadrat. However, if I notice, for example, that moss only touches, for example, one of these grids, then that would be 1 out of 100%, so 1%. So that is a quantitative survey, which gives me a much better idea of the abundances of different organisms. Okay, so that's how you use a quadrat. And I think this is where we were answering questions on yesterday. So we might just quickly go through these questions and see how you got on. So how do we define an abiotic factor? We call this a non-living factor. So e.g. would be temperature. What apparatus would you use for measuring soil and air temperature? That, of course, would be a thermometer. What would you use for pH? You would use, for example, universal indicator and a color chart, or you could say pH sensor. Light intensity, we use a light meter. Describe how to use a pooter. So for a pooter, what you would do is you put one tube over the insect, you create suction on the other side and basically inhale, and the animal is drawn into the pooter. How does a pitfall trap work? Well, a pitfall trap, you dig a hole, you put your container in, you leave it for a few hours, and the insects will fall into the trap. A sweep net. Sweep net is basically you rotate the sweep net in a figure of 8, so imagine an imaginary horizontal 8, and you sweep it through the grass, you catch the net then, and you investigate what is inside it. A beating tray, very simple, shake your bush, and then the insects fall into the bush or into the beating tray, and you just investigate them. How to use a key? A key is simple as well. So what you do is you're asked a series of questions, and you basically answer the question. So is it yes or no? Does it have 6 legs or 8 legs? And you carry on until it identifies the specific organism in front of you. What is the niche of an organism? What is the role or purpose of the organism in that environment? So e.g., a squirrel has a unique role in that it buries, we'll say, nuts, which helps trees reproduce. It's also a primary consumer. Right, so there's only one final thing here I want to mention today, and that is the Darfur scale. And this is something that we could use if we were doing a quantitative survey using a quadrat. Okay, the Darfur scale, each letter stands for a word. So that, for example, D stands for dominant, R stands for rare. So what you would do is you would come up with a scale, say, for example, if I'm going to decide if something occurs only in 2% of my survey, then that is going to be considered to be rare. Okay, if I find that something is occurring in 10% or above, maybe 10 to 15%, I will decide that that is occasional. If something, however, occurs in 90% of quadrats, then I will call that a dominant species. So in a grassland, what do we know is always going to be the dominant species? Grass. Grass is incredibly good at surviving and I suppose thriving rather in high light conditions. Okay, light intensity conditions, they are well adapted to that environment. Right, that's enough for today, I think.